Hi my dear students, very good evening all. Welcome back to Sri Ramanuja Vidya Mandir. Today I am going to explain about the how do we fall ill. The lesson name is how do we fall ill. Now in this topic we are going to discuss about the immune system and the work of antibiotics in our day-to-day life. We have discussed in the previous class, and let us review all these points. Students, first, what do you mean by immunity? It is one of the self-defense mechanism that is found in our every individual, so it protects us from the or against the pathogens. You know what you mean by pathogen. Pathogen is disease causing agent is the pathogen. Understand? Right. See here, the ability of an organism to recognize and defend itself against the specific pathogen is what? Antigens. Here the word pathogen means disease causing agent. Disease causing agent. This disease causing agent is also known as antigen. Antigen. Here now let us see about the immune response. The third line of defense involves the production of antibodies and the generation of specialized lymphocytes against the specific antigens. So students, in our blood we have three different types of the blood cells: RBC, WBC and platelets. Okay, so you know the different functions of every blood cell here. So when WBC concerned, it is taking care of the immune system. It shows immune response against the foreign pathogens. Right, now let us see about the antigen. Antigen is a molecule. It comes from the pathogen or a foreign organism, so that provokes a specific immune response. So antigen is being produced by the specific diseased microorganism. Now see, and let us talk about the antibiotics. So nowadays doctors are using antibiotics just to control the infections such as bacterial infections and all. And moreover, one important point I want to convey that these antibiotics are prepared from the microorganisms itself. Okay, so it's very important, make a note of it. Then so by in using for preparing of this my antibiotics, they'll be taking the helps of virus, bacteria and fungi. So the best example of antibiotic is penicillin, right? Okay, this penicillin is prepared from the fungi name Penicillium notatum. Penicillium notatum is the fungus name. From this fungus, so penicillin is the antibiotic is prepared. And here the first antibiotic prepared is penicillin, which developed in the year 1940s, that is especially during the Second World War time. And this antibiotic was for the first time discovered by Mr. Alexander Fleming. Alexander Fleming. Understand? Now see, there are now about nearly 50 to 100 commercially available antibiotics are there. So out of that, tetracycline, streptomycin, erythromycin are some of the best examples of generally used antibiotics. Now see, phagocytosis. This phagocytosis is nothing but the inflammation . So already I have explained about the meaning of inflammation in the previous class, I think. So the word inflammation means, students, the response, the shown by the body against the pathogen is inflammation. Actually, students, here whenever a microorganism or a pathogen enters in our body, okay, so the fate of pathogen I am going to explain now. This pathogen is going to produce this antigen. Now this antigen is responsible for the causing of disease or infection in the particular individual. Now, so this particular antigen is being immediately noticed by the WBC, and especially in the WBC we have lymphocytes. Okay, see, lymphocytes are nothing but agranulocytes, one of the type of WBC or white blood cells. This lymphocytes is going to produce antibodies against the antigen. So these antibodies are nothing but the fighters which are produced by the lymphocytes. So this antibody will react with the antigen. So obviously it will form antigen-antibody complexes. So in that reaction, our antibodies shows high response against the antigen, and it makes the particular antigen so inactive. Okay, so as a result, this inactive form of antigen unable to produce disease in the particular individual. So this is the action generally taking place in the case of antibiotic administration to the particular patient. Okay, this sort of process is known as phagocytosis. See students, here this is the granulocyte, or one of the WBC we can call. So this WBC is going to is going to eat this bacterium. So by the process of engulfing, okay, it is going to engulf the particular bacteria, so that it is going to inactivate the bacteria. So this particular technical name used for this process is called phagocytosis. So actually cell lysis will be taking place. Means cell lysis means destruction of the cell. Destruction of cell. Right? Now, so what do you mean by immune system? Immune system is a biological structures and the processes within an organism that protects against the disease by identifying the killing pathogens and tumor cells. So this is about the immune system in our body. Like other systems like nervous system, hormonal system, or blood circulatory system, in the same way we have a special system that is immune system. Like now antigen antibody reactions I just now explained, right? Actually immunity we have in our body, right? It is of two types. Okay, two types: adaptive immunity and innate immunity. Adaptive immunity which will get by the organism during its or during his lifetime. So understand, so innate immunity in the sense by birth we people will get some sort of immunity, that is innate immunity. Adaptive immunity is also known as acquired immunity. It is of two types: natural and artificial. So under natural, passive, active immunity is there. So passive means, so this will receives from the mother. So by what way? By feeding mother's milk. So in that process we are going to get some kind of antibodies. They are nothing but the immunoglobulins. So in the mother's milk we are having the very important things are known as immunoglobulins, and So this immunoglobulins is going to give the immune response or immune power which may exist in the particular individual for lifelong. Okay, so in the case of active immunity, and during his lifetime a person may get this particular thing, so known as active infection. And in the case of artificial immunity, it is again of two types, passive and the active. So antibody transfer, see, by using the antibiotics we can get this type of immunity. And here in the case of active immunization, it is also nothing but the vaccination. Okay, artificial immunity. So by using vaccines and all, generally we can increase the immune response in the particular individual. Now let us talk about the vaccination. Very, very interesting thing. So let us move on to the next slide, students. Here vaccination is defined as the protection of body from communicable diseases by the administration of some agents that mimic the microbes. So here we can protect a particular body from communicable diseases by administrating the microbes. Okay, so the agent may be suspension of killed or attenuated microbes known as vaccine. Example, let me explain, students. For example, this is the individual. You think this circle is a individual, right? Now here we have taken a microbe or microorganism, and first here they'll make the particular microbe as heat killed. Okay, this heat killed microorganism is known as attenuated microbe. Okay, though it is killed, though this microorganism is killed, but it is having the capability of what? Capability of producing infection or disease in the individual. Okay, understand? See, whenever the microorganism it is become heat killed, so we can call that as a attenuated form of microorganism. So it has the, though it is killed, so it has the capability of producing infections or causing disease in the particular individual. So whenever this human is giving a vaccine, okay, so in the form of killed microorganism or killed pathogen, killed microorganism, but our body's immune system, okay, it functions again, it thinks as a particular foreign pathogen enter in our body, so it is going to immune system responds very immediately, and it is going to start producing the antibodies against this vaccine, so that this heat killed microorganism is going to suppressed by the antibody activity which is shown by the individual. So this is the type of action will be taking place. Right now vaccines are known as dead inactive organisms. Okay, vaccines are nothing but, students, dead inactive organisms. Okay, what do you mean by vaccines, students? Vaccines are dead inactive microorganisms, or we can also call purified products derived from them. So these are several types. Okay, so maximum they are killed or attenuated. Okay, now let us describe about the vaccine now. Vaccine is a substance that is introduced into the body to prevent the disease producing produced by the certain pathogens. This vaccine consists of dead pathogen or live but attenuated artificially. So attenuated, very very important. So attenuated, right? So the vaccine induces immunity against the pathogen either by producing the antibodies or by activating the lymphocytes. See, this vaccine induces the immunity against the pathogen by producing what and what? By producing antibodies, or by activating lymphocytes. Okay, actually this is the action of vaccines in our body. So whenever they enter in our body, so it is going to activate the immunity. So activated immune power, so will produces the what? Antibodies activating lymphocytes against this. Here Edward Jenner produced first live vaccine. Who? Students, Mr. Edward Jenner produced for the first time vaccine. He produced the vaccines for smallpox from cowpox virus. Nowadays, vaccines are used to prevent many diseases like measles, mumps, poliomyelitis, tuberculosis, smallpox, rubella, yellow fever, rabies, typhoid, influenza, hepatitis B. Right? So all the different examples of viral infections. Nowadays, see, vaccines are, we are using the vaccines for all sort of these diseases like measles, mumps, poliomyelitis, tuberculosis, smallpox, rubella, yellow fever, rabies, typhoid, influenza, hepatitis B, etc. Now here let us study about the process of vaccination. So it's the process of distributing and administering the vaccine is referred to as vaccination. The process of giving or injecting vaccine to the particular child or a adult human is known as vaccination. Now let us see few more details about the vaccination. Actually, for the first time, this vaccine was discovered by Mr. Edward Jenner. Okay, he is an English doctor actually, and he will be well known as father of immunology. Jenner coined the term vaccination. It comes from the Latin word that is vaccina. Okay, it comes from the Latin word vaccina. The exact meaning of this vaccina is cowpox. Okay. Now let us see the second point, next point. See, for this experiment, he carried out experiments on 8 years old boy. His name is Mr. James Phipps. Okay, so he carried his experiments on 8-year-old boy. Name is James Phipps. And whenever vaccine enters the human body, the person, so antigen inside the body stimulates the formation of antibody. So this antigen present stimulates the formation of antibodies. So now this antibody is going to kill, or this antibody is going to inactivate the what? Inactivate the pathogen. Understand? So antibodies attack the disease causing pathogen, thus prevents its entry into the body. This is the mode of action. Mode of action of vaccine in our body. Mode of action of vaccines. So whenever vaccine is infected to the person or injected to the person, so here our body's immune system is responds against it, and it is going to produce the antigens against the particular pathogen. So this antibodies is going to kill or inactivate the pathogen. Okay, actually this is the actual procedure which involved in the vaccination. Now let us describe about what you mean by killed vaccines. What do you mean by killed vaccines? So when it is unsafe to, of course, when it is unsafe to use live microorganisms to prepare vaccines, they are killed or inactivated. So if we use live vaccines means, so of course it is going to be a problem in our body. So instead of that, they're going for killed or attenuated form of vaccines. So these are preparations of normal infectious pathogenic microorganisms that have been rendered non-pathogenic, usually by treatment with using heat, formaldehyde and gamma radiation, so that they can't replicate at all. Such killed vaccines are very greatly in their efficacy. Okay, so this is about the killed vaccine, students. Now you just see the pictures of vaccination and as well as immunization. Let us see the differences between the vaccination. Vaccination is the process of administration of antigen material to stimulate the individual's immune system to develop adaptive immunity to a pathogen. Here, in the case of immunization, is the process whereby a person is made immune or resistant to the infectious diseases, typically by the administration of vaccine. So, students, let us see. Vaccine is the administration of antigen material. Administration of antigen material to stimulate immune system to develop adaptive immunity to against the pathogen. Here, immunization process whereby a person made immune or resistant to the infectious diseases, typically by the administration of vaccines. So, of course, both will get same meaning, but slight difference is there. Now, let us discuss few viral diseases now, students. First, jaundice. It is one of the viral disease, and the disease causing agent, you know what you mean by pathogen. Pathogen means disease causing agent. Okay, and it is going to cause it by the virus name hepatitis virus. See, look at the picture, hepatitis A, B, C. There are different types of hepatitis viruses are there. A, B, C, D and E. Mode of transmission. Hepatitis A, it spreads by means of contaminated food and the water, whereas hepatitis B is by the contact with the infected body secretions like nasal secretions, tears and all, sweat and all, through all these things. So there is a chance of spreading of hepatitis B type of disease from one individual to other individual. Now, let us see the symptoms of this hepatitis, or we can also call it as jaundice, like fever and the loss of appetite and nausea and vomitings, yellowness of skin, itching of skin due to the bile pigments, urine deep yellow in color, enlarged liver, headache and joint pains. See, let us see the symptoms once again. Fever and the loss of appetite and nausea and the vomiting, yellowness of the skin, itching of the skin due to the bile pigments, urine deep yellow in color and enlarged liver, headache and the joint pains are generally observed symptoms. So, you just observe the figure, students. So, eye color will become yellow. Eye color will become yellow. Right. So, by noticing all these symptoms, we can easily came to a conclusion that particular individual is suffering with the jaundice. Now, let us see the prevention and the cure. And so, by following adequate bed rest and carbohydrates rich diet, So we have to provide the more carbohydrate rich diet to the patient, and consumption of proteins and fats should be limited. So we should particularly we should minimize the diet of proteins and the fats to the particular individual. We should encourage, or we should give the more doses of the carbohydrate rich food to the individual, and eating hygienic food and drinking disinfected water. So being in healthy and hygienic conditions, taking care of hygienic food and the water, it is the better solution for avoiding all sort of diseases, students. That's the reason elders used to say that we should, they are not encourage us to eat outside food always. Okay, our grandparents and the grandmother, grandfather, always they used to prepare the food at the home. They need to serve. It is there. They know that if we'll eat something outside, obviously we'll get some abnormality in our body. That is simply due to the they'll not maintain proper hygienic conditions. Okay, obviously they'll be use the contaminated water for the process of cooking and all. So better to avoid eating outside food. Now students, in this lesson, in this lecture, I have given the explanation about the immune system, and about the antibiotics, the process of phagocytosis I have explained, and I told you about the importance of immune system in our body, and I have explained about the antigen antibody reactions. Okay. Now, so different types of immunities we have discussed: adaptive, innate immunity. Adaptive means which is going to acquire during our lifetime, that is adaptive. We can also call it as acquired immunity. And let us start next type, it is innate immunity. So the immunity which will get from the birth itself. So, and adaptive immunity is again two types: natural and artificial. Here under natural, passive, which receives from the mother; active, artificial immunity, passive antibody transfer. Artificial means again another type is there, active immunization. And we have discussed about the vaccination also. Vaccination is nothing but it defined as a protection of the body from communicable disease by administering some agents that mimic the microbes. See, this used microbe is nothing but the vaccine. Next, I have explained the action of vaccine in our body. That vaccine are dead or inactivated microorganisms or purified products derived from them, and there are several types: skilled or attenuated forms are there. I have explained who discovered this. Mr. Edward Jenner discovered the vaccine. Okay, and so he will be treated as the father of immunology. The study of immune system or the study of immune response is known as immunology, and Jenner coined the term vaccination. This word is derived from the Latin word that is vaccina. It means cowpox. Carried experiments under 8 years old boy Mr. James Phipps. Okay, actually first he noticed that the people who are suffered with the cowpox are not getting the smallpox. Okay, then he observed all these things. So he prepared to carry the experiment on 8 years old. So the Mr. James Phipps. Okay, he given the cowpox vaccine. Okay, just he got a slight fever. After that, some time, what he did, our Mr. Edward Jenner, again he submitted the smallpox virus into the boy's body. Again, this boy does not consist of any kind of smallpox. So by observing all these things, so he came to a conclusion that cowpox, when the people are affected with the cowpox and they are not infecting with the they'll not infect with smallpox. Okay. Okay students, that's all for today. Thank you all. Thank you so much.